Welcome back, students. Today we are going to discuss part 2 of Unit 2, Unit of Life. So let's begin with the lecture. Before start the main lecture, I would like to inform that images used in our slides are for education purpose only. We respect intellectual property right of the original creator and owner of these images. If you are the owner of any of the images used in this PPT or have any concern, any issue, please contact us at sathee@iitk.ac.in and we shall remove the images. And in this particular video lecture, we are going to discuss these 2 points. Macromolecules, okay, their chemistry, cellular location, their functional signification, and some type of macromolecules. So macromolecules also comes under this particular heading, biomolecules. These are also a type of biomolecules. So what are biomolecules? Just recall firstly, biomolecules are organic compounds which form the basis of life, right? And biomolecules build up the living system, and these molecules are mainly responsible for the growth and maintenance of any living cell, right? So if we crush a living tissue, okay, if you grind a living tissue in mortar and pestle in the presence of a acid like trichloroacetic acid, and after grinding, if we strain it out, then some molecules, some part is able to dissolve in this particular acid while other is not able to dissolve. So after straining, we will get two type of fraction. One is acid soluble part or acid soluble pool, another one is acid insoluble pool. Okay, so in acid soluble pool, some molecules are able to dissolve. Those are known as micromolecules, whose molecular weight are between 18 to 800 Dalton, like minerals, sugar, gases, water, some amino acid, nucleotides. Okay, while in acid insoluble pool, all those molecules whose molecular weight more than 10,000 Dalton, whose molecular weight more than 10,000 Dalton are present in acid insoluble pool, like carbohydrate, protein, nucleic acid and lipid. But this lipid is a type of exception. It's a type of exception. This is not truly a macromolecule because its molecular weight is around 800 Dalton. So it's a small molecule. But during grinding, lipids are present in the cell membrane as well. Okay, so during glycolipid is there. Okay, it's a part of cell membrane. So during grinding, what will happen? This lipid that cell membrane disrupt, and this lipid form vesicle, and the vesicle size is comparatively large. So this lipid molecule is not able to cross that sieve. Okay, so it is found in this fraction only, acid insoluble fraction only, but it is not a truly macromolecule. Okay, so all the molecules which are present except lipid, as I told you, which are present in acid insoluble pool comes under this macromolecule category, while all those which are able to dissolve in acid, this trichloroacetic acid, that all type of molecule comes under micromolecule category. Okay, now so let's discuss macromolecules. So the acid insoluble fraction, just now I have discussed this acid insoluble fraction. So in this acid insoluble fraction, four type of organic compounds we can analyze, like protein, nucleic acid, polysaccharide and lipid. But this one is exceptional. I have mentioned it over here. Okay, so these classes of compound have molecular weight in the range of 10,000 Dalton and above. Exception lipid, because lipid's molecular weight only 800, approximately 800 Dalton, very less. Okay, so lipid are indeed small molecular weight compound and are present not only as such but also arranged in the structure like cell membrane and other membrane, and because of the disruption of cell membrane it will able to form vesicle, and these vesicles are not able to cross the sieve. That's why it is found with the some other macromolecule content, right? Okay, so what is the average composition of the cell? So if we calculate or analyze average composition of the cell, then we can find out that the maximum percentage water, you can see over here 70 to 90% water is present inside a cell, inside the cellular mass, and after that protein 10 to 15%, and just after this protein, nucleic acid is there 5 to 7%, followed by carbohydrate, then lipid, and then some ions. Okay, so maximum water, and then just after the water, next category protein, this one protein. Clear? Okay. Now, so in today's lecture, we are going to discuss macromolecules. So first type of macromolecule is polysaccharide. Okay, so these polysaccharides are long chain of sugar. These are made up of a small unit. A small unit of, what is the smaller unit of basic unit of this saccharides? These are monosaccharides, right? So they contain different monosaccharide as a building block, right? So they contain different monosaccharides as building block. Cellulose is a polymeric polysaccharide, consists of only one type of monosaccharide, that is glucose. So it is a homopolymer. If only one type of monosaccharide is present in that long chain, then that type we can use the term homopolymer, and cellulose is one of the example of homopolymer, right? Starch is another variant of this, and as you know very well that plants store its own food in the form of starch, right? So starch is also known as a storehouse of energy in plant tissue, while in animals have another variant. What is the name of that variant? This is glycogen. Animals store their food as a glycogen, right? So these are certain examples of polysaccharide, and these polysaccharides are not sweet in taste. These are not sweet in taste, hence they are also called non-sugar. While if we are compare this polysaccharide with monosaccharides, some monosaccharides are sweet in taste like glucose, fructose, not all. Okay, so but polysaccharides are not sweet in taste, so that's why we can use the term non-sugars for them, right? They are amorphous in nature and insoluble in water, right? In polysaccharide, the individual monosaccharide are linked together by glycosidic bond. Okay, and this bond is formed by the dehydration. If this is one type of monosaccharide, another type of monosaccharide, so after dehydration, the bond formed between these two is known as glycosidic bond. Okay, yes. So that's why I have written over here that the bond between two monosaccharides while forming polysaccharide, what kind of bond is this? This is glycosidic bond. Okay, so this bond is formed between two carbon atom of two adjacent monosaccharides. In polysaccharide chain, the right end is called reducing end. If we see whole structure, whole biochemical structure of this polysaccharide, that time right end is called reducing end, while left end is called non-reducing end. Okay, clear? Now, plant cell wall, these are also made up of cellulose. Certain kind of products like paper, cotton fiber, they all are cellulosic. Means these all are polysaccharide in nature. Okay. So, and apart from this, some other modified sugars, modified amino sugars, they also combined with the polysaccharide and form different type of structure. That's why it is written over here that there are more complex polysaccharide in nature. They have a building blocks of amino sugar and chemically modified sugar like glucosamine and acetylglucosamine. Okay, these are certain examples of chemically modified sugar or amino sugar. Similarly, some exoskeleton of some arthropods. These are also made up of polysaccharide, and that polysaccharide is known as chitin. Okay, and this chitin is almost also a homopolymer. Okay, so there are few examples like cellulose, like chitin, and like different type of glucosamine and glucosamine. So these are the examples of polysaccharides. Now let's discuss second macromolecule type, that is protein. Okay, so proteins are polypeptide. These are polypeptide. Each protein is a polymer of its micromolecule or its monomer unit. It's a polymer of its monomer unit. And what is the monomer unit of protein? Or it is amino acid. Okay, so each protein is a polymer of amino acid which linked by a specific bond, special bond known as peptide bond. What is the type of bond? Peptide bond. As in the case of polysaccharide, that this bond is when two monomer units combine together, carbon carbon. What is that bond? That is glycosidic bond in the case of polysaccharide. But here, if two amino acids combine together and form a bond, this type of bond is known as peptide bond. Okay, as there are 20 type of amino acids, like there are certain examples: alanine, cysteine, proline, tryptophan, lysine. There are certain examples. Okay, a protein is a heteropolymer and not a homopolymer. Means in a protein there are combination of various amino acids. Okay, there is a alanine is there, cysteine is there, proline is there. So there are combination of various amino acids. That's why it is written over here that protein is a heteropolymer. It is not a homopolymer. Okay, certain amino acids are essential for our health, right? And they have supplied with our diet. Some amino acids are very, very important. So we have to take all those amino acids through our diet. Okay, so dietary protein are the source of essential amino acids. So we have to consume this essential amino acids through our diet. And essential means some are non-essential also present, right? But our body can make those non-essential. That's why it is written over here that this non-essential our body can make. Okay, only those comes under essential category for which our body is not able to synthesize that one. Okay, we have to take it from the outside with our foods, with our dietary habits. Okay, now there are plenty of work has been done by different type of protein. As you can see, protein can act as a intracellular substance, protein can act as a enzyme, protein can act as a hormone, protein can act as a antibody in our defense system. It is also present in a certain kind of receptors. It may be sensory receptor or other kind of receptor. They can also act as a transporter. Okay, they can used up in transport system to transport glucose. So this is the list which is given in your NCERT biology book, and this is how you can now understand different kind of function of protein. Okay, and out of this whole list, this collagen one is the most abundant protein in the animal world. Collagen is the most abundant protein in the animal world, while this rubisco, ribulose, biphosphate, carboxylase, oxygenase, this enzyme, this particular protein is the most abundant protein in the whole biosphere. This is again very important. So if the question asked about biosphere, we have to take rubisco. If question asked about only in animal world, then we have to take collagen. Okay, very frequently questions related to these two lines comes in different competitive examination. Now, structure of protein. The primary structure of protein refer to the sequence of amino acids. Sequence of amino acid, that is known as primary structure, just a polypeptide chain. Okay, so the primary structure is held together by peptide bond that are made during the process of protein synthesis. As you can see in this particular diagram, serine is there, cysteine is there, tyrosine, glutamine. So this is the primary structure, just alignment of different amino acids. Okay, so the first amino acid is known as N-terminal amino acid, and the last one is known as C-terminal amino acid. So this is the primary structure. Just sequence of amino acid in a polypeptide chain is known as primary structure. Right, clear? Now, secondary structure. Secondary structure refers to the highly regular local substructure. This is local substructure on the actual polypeptide backbone. There are two different type of structure. One is alpha helix and second is beta strand or beta sheet. They both are different type of secondary structure. Okay, and the tertiary structure refers to the three-dimensional structure, and this one is created by a single protein molecule, and it may include one or several domains, but only single protein molecule. This is important. So the alpha helix and beta plated sheet are fold together and form a compact structure, a globular structure, and this folding and unfolding, this is driven by some hydrophobic interaction. What kind of bond is there? Hydrophobic interaction. And this folding, after the folding of beta plated sheet, alpha helix, they are able to create tertiary structure, right? So tertiary structure is absolutely necessary for the portions of protein thread are arranged in the form of helix, right? So this one is the secondary structure. After again recoiling of this secondary structure will form tertiary structure, and in this tertiary structure both the type of alpha helix and beta plated sheet are there. Okay, and what type of interaction is there? Hydrophobic interaction, right? And after tertiary structure, final type of structure is known as quaternary structure. This is again three-dimensional structure, and it consists of aggregation of two or more individual polypeptide chain. So here two or more polypeptide chain now onwards. Okay, and this polypeptide chain operate a single functional unit, and now this polypeptide chain operate as a single functional unit, right, like hemoglobin. Okay, so the resulting multimer is stabilized by some non-covalent interaction and disulfide bond as in tertiary structure. Okay, as in tertiary structure. So what kind of interaction is there in secondary one? Hydrophobic interaction. What kind of interaction is there in tertiary structure? The non-covalent interaction and disulfide sulfide. What kind of interaction is there in quaternary structure? Non-covalent interaction and disulfide bonds. And in quaternary structure, 2 or more individual polypeptides are there. Okay. In secondary structure, only one type of polypeptide chain are there. Right. Now you can compare all type of structures by seeing this diagram. Just alignment, amino acid alignment in a polypeptide chain. Methylene, phenylalanine, asparagine, alanine, valine, leucine and glycine and so on. This is known as just alignment of amino acids. This is known as primary structure. In secondary, it fold into different orientation like beta sheet or in alpha sheet. This one is known as secondary structure. Okay, and tertiary structure, refolding of this both the forms of secondary structure make tertiary structure, and this time we can use the term three-dimensional structure we can see. Okay, but only one type of polypeptide. But after the folding of tertiary structure, this quaternary one is formed, and in this quaternary form more than one monomer unit are there. Okay, so this is the structure of quaternary one. Now next type of macromolecule is nucleic acid. Okay, so this nucleic acid are the complex long chain compound that contain carbon, oxygen, hydrogen, nitrogen and phosphorus. And these nucleic acid are the basis of genetic material of all organisms. Okay, what is the simplest unit of nucleic acid? It is nucleotide, and this nucleotide we have already discussed in micromolecule level. When we discussed in last lecture micromolecule, we have discussed this nucleotide, right? And this nucleotide is made up of 3 different things: a kind of sugar that may be pentose or pentose, but ribose or deoxyribose show a kind of sugar, a base, this base is nitrogenous base, and a phosphate group, right? So sugar plus base plus phosphate group. Okay, so these 3 are the components of nucleotide. If you discuss about different type of nitrogen base, then we can categorize bases into 2 different category: purine and pyrimidine. Purine base comprises mainly adenine and guanine, both are double ring structure, right? And pyrimidine, cytosine, thymine and uracil, these are single ring structure. Okay, in RNA, in place of thymine, uracil is there. Okay, so in nucleoside, what is the difference between this nucleoside and nucleotide? Nucleoside and nucleotide. Nucleoside is made up of base plus sugar, and if phosphate is also attached with this base or sugar, or collectively we can use the term for both of them nucleoside, then it will convert into nucleotide. Okay, so after the attaching of this phosphoric acid or phosphate group, we can use the term nucleotide. Clear? A nucleic acid contains deoxyribose sugar. So deoxyribonucleic acid, DNA, while ribose means ribonucleic acid. Ribose is a type of sugar which is present in RNA. Deoxyribose is a type of sugar which is present in DNA. Right? Now this is the double helical structure of DNA. As you can see, this sugar, this sky blue part is sugar phosphate backbone. Sugar phosphate backbone. And if you compare this sugar phosphate backbone, you can see over here, this one is sugar, deoxyribose, phosphate, this sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. So collectively we can say the term sugar phosphate backbone, right? Sugar phosphate backbone, right? Sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, like this. And all the bases These are present just on the right angle, you can say, and inside the chain. And these bases, adenine, thymine, guanine, cytosine, with their respective base, they can form hydrogen bond. As you can see over here, within cytosine and guanine, 3 hydrogen bonds are there. 3 hydrogen bonds are there, and adenine and thymine, only 2 hydrogen bonds are there. Okay. So, and this model double helical model of DNA is proposed by, this was proposed by Watson and Crick, and for this very, very important work, they also got Nobel Prize for the same. And this model says that DNA exists as a double helix. Because in very detail about this model, what kind of work they have done, what other scientists also involved in this particular world who have done that X-ray crystallography, that we will discuss in genetics part. Right now we are in macromolecular molecule chapter, so just we have to understand the type of nucleotide, nucleoside, and total nucleic acid, right? So this model says that DNA exists as a double helix. The two strands of polynucleotides are anti-parallel. Polynucleotides are anti-parallel, means they run in opposite side. If first one is run like this, then another like this, 5 prime end over here and 3 prime end present over top side. Okay, and the backbone is formed by sugar phosphate sugar chain. Just now we have discussed this. The nitrogen base are projected more or less perpendicular to its backbone but face inside. Just now we have discussed this point also. Adenine and guanine of what stand compulsory base pair with thymine and cytosine respectively. Okay, adenine always pair with thymine and cytosine always pair with guanine. Clear? Now, so this is the double helical structure. Okay, there are 2 hydrogen bonds are present between adenine and thymine, and 3 hydrogen bonds are there in guanine and cytosine. And each strand appear like a helical staircase. You can see helical staircase, right? At each step of ascent, the strand turn 36 degree. 36 degree. Okay, the pitch would be 34 degree, and the rise per base pair, means distance between these two base pair is, as you can see over here, the distance between this and this one is 3.4 angstrom, and this type of DNA model is called B type of DNA. Okay, so this is all about the macromolecular part of biomolecule chapter. If you now if you have any question, any doubt related to the today's topic, you can write down to me comment box. We will discuss that particular topic later on. Okay, till then, take care. Bye bye. Namaskar.